Well, hello, my name is Cara Santelli. I'm a curator in the Department of Mineral Sciences. And for those of you who are always unsure as what to introduce me as, I'll just let you know that I consider myself a geomicrobiologist, but of course you're all free to call me whatever you like, especially Dr. Samper. But as a geomicrobiologist, I'm interested in understanding the impact of mineral-microbe interactions. Specifically, I study the process of biomineralization, so the process by which microorganisms, living microorganisms, actually form or precipitate minerals, such as you can see here, this fungal cell precipitating the manganese oxide mineral on the outside. In addition to forming minerals, microbes have a very important role for dissolving minerals or mineral weathering, such as forming soil formations, such as you see here. And both of these processes are very important for controlling the distribution and the concentration of essentially every single element in nature. And having a better understanding of these processes can help us apply our knowledge to a wide variety of other disciplines. For example, in terms of improving biotechnology, improving bioremediation strategies for polluted environments, metal polluted environments, such as this environment here, uranium and contaminated environment. We can understand how to better preserve some of our cultural heritage sites, especially some of our stone sites. In addition, we could potentially even learn something about how fossils are preserved in the rock record. And for the past several years, I've been understanding mineral microbe interactions in the context of manganese, the metal manganese. And manganese is an essential element for life. Every single organism, every single living organism needs some trace amounts of manganese. But in certain environments, it can actually build up to very high concentrations, and it's a priority pollutant, such as you see here. This is a big problem in Appalachia. All throughout Appalachia, you have literally thousands of abandoned coal mines generating acid mine drainage, so these low pH waters that are just chock full of dissolved metals. And what we know so far is that bacteria, and what we're learning now, that fungi are actually the important drivers in controlling the distribution and the concentration of manganese in the environment. And they do this mainly by one process. So they control actually the valent state of manganese, or the oxidation state of manganese. So in these waters, in these contaminated waters, manganese is soluble, it's dissolved, and it exists in the manganese 2+ form. But it turns out that microorganisms can induce an oxidation chemical reaction. And what they do is they convert manganese 2 to manganese 4+. Manganese 4 is insoluble. It precipitates out as a mineral form. And this is a very important process for cleaning up some of these contaminated environments. So this is actually a biomineralization process, converting the soluble to an insoluble form. So one of my primary objectives is to determine who are the primary drivers of manganese biomineralization in the environment, particularly in some of these really polluted environments. And so I started with a culture-based approach. I actually went to the environment and tried to figure out which organisms could oxidize manganese, could precipitate these manganese oxides, and which couldn't. And so what I found was really surprising. We were really expecting to find mostly bacteria in these studies, and what we found, and when we went to these really polluted sites, we found mostly fungi that were actually able to precipitate the manganese oxide. So here what you see are 3 different species, and they're growing from a central point. They grow radially outwards, and that brown color that you see is not a pigment, it's actually the precipitation of the manganese minerals. So they're normally clear. If I didn't have any manganese in solution as they were growing, they'd be completely clear, they would grow. But as they're growing here, in the presence of manganese, they're precipitating this mineral. But culture-based approaches have severe limitations in terms of understanding really who are the most important organisms in these environments for promoting manganese biomineralization. So right now we're going to pursue some molecular approaches, some more quantitative approaches, and this is work being pursued right now primarily by a postdoc in my lab, Dominic Chaput, who I'm happy to report just received the Secretary's Distinguished Research Fellowship. And we've received a number of different funds internally to sort of get at who are the important drivers using some of these more molecular approaches. In addition to studying the organisms, I'm really interested in what they are producing. What is the byproduct of this mineralization product? There are over 30 known different manganese oxide minerals that can exist in an environment. They tend to be very poorly crystalline, and they tend to be very small. So think nanocrystalline size or microcrystalline size. And they're very important for the general health of the environment. So in addition to microbes influencing the general health of an environment, these minerals also influence the general health of an environment. They are often called the sponges of the sea. They can absorb out all sorts of nutrients and metals. And so what we found from our initial studies is that all the fungi, at least, seem to be producing a mineral called bernacite. Here's a structural model of the mineral bernacite. But each species has very small variations in the structure, and this could have huge relevance for how important these minerals are in the environment. And this is some research I'm going to be pursuing this summer in collaboration with Dr. Post, the head curator of the mineral sciences, and a summer intern. So stay tuned for some of our results. But for one take-home message, the one take-home message is that our results indicate that fungi are actually, at least in this environment, maybe even more important than bacteria in driving some of these biogeochemical cycles. And I don't think my results are very unique. I think as the more we look, the more we're going to find that fungi are actually very important for the biogeochemical cycling of a lot of these different elements, maybe even being used for a cleanup of some of these contaminated environments, including asbestos contamination or uranium contamination. So I just want to leave you with that, and here are my, thank you, all my collaborators and my funding.
Will you tell us in words of one syllable what your work could produce in terms of cleaning up the environment?
So the question is what the microbes could produce?
Well, the fungi or whatever you want to call it.
Yep, could produce that could clean up the environment.
Yes.
So in terms of cleaning up the environment, the actual production of the mineral phase is what's most important. So basically they're taking the dissolved metals out of solution and forming something insoluble that can easily be stripped out, especially in various technological advances or just naturally in the environment. You have these minerals they'll settle out of the environment and take that dissolved form out of the water system, and this is really what the process is. But important for the manganese oxides, if you can produce manganese oxide minerals, the interesting thing about those minerals is they, like I mentioned, they absorb all sorts of other things. And so there are other elements like other metals such as zinc and lead and copper in some of these environments. And so if you can get the manganese oxide to form, you're essentially killing two birds with one stone, because
How do we do that? What do we do?
So, well, we need to understand the microbes, first of all, that are primarily promoting this process. We don't really have an understanding of how they do it. So one of the things I'm working on right now is figuring out the mechanism. Is it an enzymatic mechanism? Is it a direct mechanism? Is it something indirect? What we're finding out right now is that surprisingly, it's been really hard to figure out how they actually do this, the microbes. And what we think right now is some of the microbes are producing a chemical, and that chemical is then interacting with the manganese. So it's a very indirect process. It's very difficult to study. It's very difficult to track using genetic markers. And so we're trying to figure that out right now. Growing these things in the lab. I have the isolates now. We can grow them in the lab. We can test to see, are they phosphorus limited in their growth? What do they need in order to grow better, in order to produce these minerals better? So those are some of the things that I'm working on right now.